Today's presentation takes about 25 minutes. I'm going to briefly talk about the background of bioelectronica and an AI-driven culturomics platform called Smart Culture X. Then we're going to dive into more details on Hypercell, the high-throughput single-cell sorter, and Apfinder AI, the bioinformatics tool we developed to rapidly analyze antibody sequence. The last part is the customer map we have all over the world. Bioelectronica is a world-leading company on developing useful tools for biotech industry. By integrating microfluidics, optics, and AI technology in order to help the biotech industry to accelerate the drug discovery journey. The company started on 2015 at Reno, Nevada. On 2024, the company got successfully commercialized Hypercell and the Apfinder AI system. So far, it's been half year. The innovative AI-driven high-throughput culturomics platform called Smart Culture X is primarily designed for microbiome research due to a lack of tools for culturing bacteria strains and a need for anaerobic solution. We have developed this culturomic methodology that uses a wide range of conditions to isolate and characterize microbial taxa. This approach combines computer vision system and machine learning to monitor the bacteria growth and perform the strain clustering. We precisely control the culture conditions through a stringent rule and utilize our proprietary AI spectral algorithm with streamlined workflow to significantly reduce the hands-on time for isolation. The purpose of doing this is to improve the efficiency for rare microbial identification. Please let us know if you would like to execute some isolation of the microbiome or other applications. We would like to collaborate and explore more interesting research. Let's change the world. Now it is the time, Hypercell. Before I start, could you please type yes if you work on antibody discovery yourself? Thank you. I just want to know how many of you guys directly work on this field. Okay, I saw one. There's only one person work on antibody discovery. Okay. Oh, I'm sorry. Could you please mute yourself? There are limitations of each antibody discovery technology. Hybridoma is limited by the low fusion efficiency, genetic instability, and the time-consuming nature. Single B technology is limited by isolating and cultivating B cells and the lack of robustness for screening. The display technology face issues with biased repertoires, potential of losing antibody pairs, or misfolding of the displayed proteins. We want to improve the efficiency for antibody discovery process with a small footprint, user-friendly equipment. Requires only minimum hands-on time. As you can see, the Hypercell is a microwave-size instrument, perfectly fit on your benchtop. The reagent we provide, including the sorting kit and chips. The binding principle is we generate PAWS, the polydispersed obligate dispersion system, by vertexing the oil phase and the aqueous phase at a certain speed. The PAWS capture plasma B cells, secrete antigen-specific antibody. There is an aggregate. The PAWS capture plasma B cells, antigen Bs, and IgG Bs. If the B cells secrete antigen-specific antibody, there is aggregation formed, as you can see on the right side of the screen. If the B cells is a low secretor or non-secretor, there is no aggregation formed. This approach can be directly applied on T cell or other cell type secretion profiling. For example, the Bs can form aggregates when a TNF alpha presents and sort the functional T cells out for cell therapy validation or production. Again, please let us know if you're interested in this type of screening. We would like to collaborate on it for a gene cell therapy. The Hypercell platform integrates computer vision system in sorting. The beads cluster found near the high secreting cells can be detected by a computer vision system, as shown in the rectangle. Once the aggregation detected, it triggers an electricity shock to change the size of the pods, and the bigger pods are directed to the hit collection outlet. The workflow of Hypercell is first sacrifice the mice or collect a rabbit PBMC and do a B cell enrichment. For rabbit, there is one more step of activation and culturing. Then mix the enriched B cells in aqueous phase with oil phase, then vertex. After the incubation, quickly observe the aggregates under microscopy and the cells are ready for sorting. The sorting step can take up to 70 minutes, followed by a quick recovery step. The cells are ready for sequencing. After getting sequences from the vendor, the customer can analyze data with ApeFinder and choose the best cell sequences, antibody sequences for validation. Okay, a quick summary of the technique. Hypercell combined fusion, subcloning, and screening steps in sorting. Save customers at least 4 to 6 weeks compared with hybridoma. We can easily apply this workflow with multiple types of antigens across the species for antibody discovery application and also for sequence-based cell sorting, which is a mission impossible for flow cytometry sorting. Hypercell is much gentler methodology which can keep the best cell viability and the BCR diversity. It is easy and more efficient compared with the other technologies. Previously, I mentioned the advantages of Hypercell compared with hybridoma and flow sorting. Compared with Beacon, Hypercell is 10 times higher throughput with 10 times reduced cost. Compared with Cytomine, Hypercell is also cost and user-friendly. The next part is ApeFinder AI, a proprietary analysis pipeline specifically designed for single-cell BCR sequencing data analysis. Could you please tap on the screen and tell me what pipeline you are using right now for antibody sequence analysis, if it's from your own or from another company? Thank you. The BCR repertoire is incredibly complicated due to the random recombination of VDJ gene segments, somatic hypermutation, and insertion, deletion, and VDJ junctions. ApeFinder AI is specifically designed to handle this complexity and provide a streamlined and accurate analysis process. ApeFinder AI integrated advanced bioinformatics pipeline to accelerate NGS sequence analysis. It enables scientists to rapidly analyze antibody sequence with minimum effort. The AI-powered workflow enables automatically identified hits antibody. This system keeps improving from a large data set over time, significantly reduce the hands-on time with improving of the accuracy. Customers can process enriched B cells from Hypercell or from any other single B cell sorting. After single cell sequence, upload the data to ApeFinder AI. It will do data analysis, provide hits and recommendations based on the antibody sequences and docking, then execute downstream validation. In order to provide flexible deployment options, ApeFinder AI is available on cloud and on premises for customer to choose, which can safely guard unauthorized access. ApeFinder AI is an automated workflow to annotate CDRs with gene, J-gene, pairing VH and VL, detect the liabilities in CDR regions, identify clonal expansion, somatic hypermutation, and quantified repertoire diversity. Graphically, analyze the V gene usage, CDR length distribution, and clone type diversity. ApeFinder AI utilizes barcoding system to filter out abnormal sequences and perform VHVL pairing. It uses IMGT database to number schemes and annotate CDRs with gene J-gene of VHVL, then present the feature of VH with gene somatic hypermutation CDR length, identify liability of antibody sequences. The tool also provides a visualized chart for the customers to explore the BCR repertoire characteristics, such as V gene usage, CDR distribution, clone type size. Then the system will group the antibodies based on the sequence similarity, CDR clone types, and structural features, with employed AI-driven hit expansion from different groups to know where the binder exists and expand the diversified candidate pool. By applying Z-score standardization, each indicator's value is transformed into a relative position measured in standard deviations, allowing for a unified comparison of different indicators in subsequent analysis. We recently updated the docking function as well. First, homology modeling and AI method are involved to predict and optimize the 3D structure of antibody to improve the accuracy and stability. Apply the protein 3D structure predictions to simulate binding interaction between antibody and antigen in order to predict the binding affinity and provide the basis for antibody selection. Here we summarize the analysis pipeline. First, we digest with 10x data, pre-processing data with remove chaotic, not fluent, or non-functional. Then we annotate the data with CDR, VDJ genes, clustering them based on VH, CDR, and clone type, and then feature the sequences based on those characters. Finally, we display the results based on a bunch of the characteristics of the antibody. The VHH sequence analysis pipeline is pretty similar to the IgG ones. Again, we use NGS data with a UMI barcoding, then pre-process to remove the unfunctional sequences, annotate based on CDR and VDJ gene, filtering the unfunctional sequences, clustering them with VH, CDR, and clone type, and finally feature them and do a result display. Customers can utilize the analyzed antibody sequence with antigen structure provided from PDB database, with a structure simulation step to perform the docking function by using comprehensive docking tools with multiple scoring system, then normalize and weight the scores, finally provide a docking score for future minimized customers' hands-on time. We have some customer studies to share with you all. In the first case study, the customer immunized mice with MHC class 1 molecule and finally got 70K B cells for sorting. We generate 536 sequences, then they selected out 62 to validate, and finally they got 42 binders. By the side, there are visualized chart from ApeFinder AI. The binding ratio from this study is 67. By further apply the docking function in ApeFinder AI, we can easily improve the binding ratio up to 77. By the side is evolutionary information to help choose the candidates for validation. The ELISA results indicating the binders from unpurified supernatant. From purified monoclonal antibody, all the binder curves are tight and neat. The BLI study showed binders KD are sub-nanomolar level. In a cohort study, the customers immunized the second mice parallel to confirm the reproducibility and consistency of the instrument. After applying the docking function, the binding ratio is very similar to the previous study. In the second case study, they immunized the mice with a surface antigen of hepatitis B virus. They got 400K B cells, and finally they got 2,500 candidates. They select 34 for validation, and finally they got 20 binders. The binding ratio is approximately 60. Here is ELISA binding results. In this slide, it shows the ELISA binding results and the BLI binding kinetics, which is also sub-nanomolar level. In the third case study, they immunized a rabbit against HB antigen. They got 500K B cells, select this much of the candidates, and finally they got 24 binders. They confirmed the binding ratio with ELISA results. The binding ratio is 100%, which is excellent. We have a lot of customers all over the world since last year. Please keep following on our LinkedIn. There will be more case study such as transmembrane protein and peptide applications published very soon. And also check on our website. Please feel free to apply AppFinder AI for a free trial today. Thank you.